Hi, this is Dr. Yunlong Liu, and this is the second video talking about how to visualize your own data using the UCSC Genome Browser. Let's go there. We'll type in genome.ucsc.edu, and again, we'll go directly into the HD38 genome build. And then sometimes you see this will happen, and what you can do is very easy. You just show the default tracks. Click this button, show the default tracks, and all the default tracks will be listed here. And you can customize the way you like and select the ones that you think is more useful. Okay, so the data I'm going to show you today is some of the data we generated in our own laboratory. It is a TAC-seq data set. So in the future modules, we'll talk about the biochemistry part and data analysis element of a TAC-seq. But today, I want to focus on visualize this TAC-seq data using the Genome Browser. So before we load our data into the Genome Browser or visualize using Genome Browser, and our information already processed the data and generated a BAM file and another file format called the big Vigo file. And that is a binary format that is very, very large. It's indexed, but it tells you where the biological signals are. So I will show you how to paste and put these files into the Genome Browser to visualize that. As I mentioned earlier in the second, in one of the lectures talking about the different ways of putting the data into the Genome Browser. And this is the second element, the second part of it, which is you put your own data actually in one of our HTTP servers, and then you only put the link of this data set, send that to the UCSC Genome Browser. When you visualize your data, you send a genomic coordinate you want to look at, and then you send this information to the Genome Browser. Genome Browser is going to place that query and send a request to the server in our institution, which is the HTTP server, and bring that data back in and put in this visualize mode and then show up in your computer. So the data itself is still in our own server, only the link are being sent to the UCSC Genome Browser server. Okay, in terms of operating that, it's pretty similar to what we did last time. You still click this Add Custom Tracks. And after you do that, it take a minute to load. And then you come to this page that we have seen last time. So now let's come in back to the file I prepared here, which is the BAM file example. So let's take a look at the BAM first. As you can see here is there's a track information, track type is the BAM, and there's the name of the track, there's a description of the track. Now you're seeing this is a big data URL, and which is a link to our HTTP server that is in our institution. So you can see that we have a server, HTTP server called Clark, and then we create a folder called NGS2, and there is a specific folder for this demo. It's a full demo. So you can see this is the file name after that. And the HTTP server is again in our institution here. So what we need to do is we will paste and copy this into this URL, this box, and then we do submit. So now instead of the data, which is hundreds of giga in size, being sent to UCSC, only the link are being sent to them. Now you're seeing there are two tracks. One is control, one is treatment. So let's click go. So now what you are seeing is that there is two more tracks that are being shown here. There are very dense signals around here. So I really, honestly, I cannot see anything. All right, so let's move this mouse a little bit. We come to this custom tracks session. So you can see both of these two tracks are being demonstrated in dense. What you can do is let's show them in full to see what happens. Again, you can play with it and then to see what different mode will give you in terms of the appearance. So now let's click this refresh. So now you can see that there is a lot of different segments here, and each one is actually a sequencing raise. They're too much. So let's come back to this. Probably full is not the best viewing mode for this. Let's change this to squish. So let's go there and click this refresh. Okay, so now you see those sequencing rates are being aligned in this particular way, and then it's a little bit more manageable. So the other tracks are still being here. So now you can see there are some of the little dashes, and each dash, as I mentioned earlier, is one sequencing race. Now you can zoom in and zoom out to look at the details. So let me do this. We can zoom in from here to here, just drag your mouse over here, and then it will come up this particular window. You click zoom in. You can do other things. You can feel free to read it. But now you see, once we zoom in, you are seeing all these different bigger dashes. There are different colors. Red means this particular read is on the positive strand. Blue means it's in negative strand. Okay, this is where the sequencing reads are. Remember that when we did NGX experiment, we got millions of different reads. Each individual one are being aligned back to the genome and created this BAM file. So each individual row is a read. So this each individual dash here is one row in the BAD file that tells you where those sequencing reads are. Okay, now just refresh your memory. UCSC doesn't have our data. Our data is in our server. So once I drag the mouse there, actually that send the genomic coordinate I'm interested in to the UCSC, and the UCSC server is going to query my server in IU, and then send this chunk data back to the UCSC Genome Browser server, and then we are viewing from there. Okay, so let's click one of these reads. Actually, it's clickable. Let's do this. So now I'm clicking one read here. So now you see that most of the information in the BAM file I demonstrated in the earlier and are being shown here. This is read name, the genomic coordinates, and the other information. And then you can see this particular cigar strain, which is in this case is 50M. Remember, 50M means 50 base. They are perfectly matched or mismatched. And there's insertion deletions, and it depends on different rates. You potentially will see that information. Now you are seeing that here is the sequencing rate. This is our rate, and this is the nucleotide sequences on the reference genome. And in this case, it's a 50 base perfect match. And bottom here is the quality score for each individual nucleotide. Remember, this is the Fred score calculation. If it's a 30, that means you have 0.1% of the chance making a mistake, and which most of these rates are pretty accurate. All right, let's get back. And then let's zoom out. Take a look at a little bit higher level. Further zoom out. You can always do this. So now each time you zoom out, you will be able to see a little bit like a broader region, so that each individual signal becomes smaller and smaller. Well, now what we are seeing here is something become interesting. Clearly, we see a lot more signal in these regions than these regions. I don't know what's going on. So let's zoom out a little bit. I still don't want to take a look at. I mean, off the script right now. I just randomly seeing what we can take a look at. Okay, so now I'm seeing there are clearly two peaks here. I don't know what they are, so let's go there. Zoom in this region, and mouse over and drag it so I can zoom in this region. So now you can see there are many sequencing rates. There are positive strand, there are negative strand rates represented by different colors. If you further look at this, okay, and there is something that is weird here. So you can see this line is more colorful than other regions. I want to see what's going on here. Let's go there. I'm zooming in here, and I click zoom in. Wow. Now you are seeing in this location, once you zoom in enough, so you can see your reference genome are being shown here. And then these are the nucleotide genome that is potentially different or similar from the reference genome. So let's go one of those. So I think this is a different one. So let's further zoom in. Let me see whether I can see it very clearly. I may not. I may already have zoomed in enough. So let's click one of this read here. See what's going on. So as I mentioned earlier, that you can see that all those information, the read name, genomic loci, and everything is showing here. In this case, it's the 50M, a 50 base match or mismatch. Okay, so now let's take a look at this particular one. It's slightly different from the previous one. So the top line here is still the reference genome sequence, and it's our sequencing rate sequence. And the bottom one is the reference genome sequence. Did you see this? So everything is a perfect match, C with C, A with A, but here, our rate is a G, reference genome is A. Clearly, for this individual, there is a genomic mutation in this region. At least in this one, for this read, I detect something different from the reference genome. I have no idea whether this is a mutation or genome mutation, or this is just sequencing error. It could be sequencing error. But once I go back here, and I'm seeing that many reads here that suggesting it's a reference genome. There are some of them suggesting it's not an alternative allele. So when you don't see this particular like a letter, which is G, and that is actually agree with the reference gene, which is A. So I don't know what the variant variant color will tell me, but for me, this is very much like a heterozygous variant locations. That's what I meant, that you can potentially, using this tool, to really go back to your data to identify genomic mutations. Of course, that ATAC-seq is not specifically designed to identify genomic mutations. And but nevertheless, we can see it. And if it's a DNA sequencing, if someone tells you there is a heterozygous variance in this location, you can potentially go there, take a look. Okay, so that's the point I want to make for this part. Now let's go back. So let me zoom out to make the signal a little bit more manageable. And similarly, this is a BAM file. Now let me put in the Vigo file here. Paste and copy here. I will go to this manage custom tracks, and I would leave them there, and now I will add additional custom tracks, and I paste and copy here. It's being processed. So now you can see there are 4 tracks, and 2 of them are Vigo files, 2 of them are BAM files. So let's take a look at what they show in the Genome Browser tracks. Again, these 2 BAM files are there, but if you look at it carefully, there is 2 more tracks are being shown here. One is here, the other one is here. So let's go to these custom tracks. So if you look at it, this is the BAM file, this is the BAM file that they were ordered based on their, I assume this is alphabetically ordered based on the track name. But if you can change this big wig file into a full rather than using as dense, let's go back here. So now you are seeing some of the information like this. It's very much like a Vigo file, which means I'm not only interested in start and end location, but for each individual segmentation, I want to see how many, what are the signals? It can be a continuous signals that at this particular location. Okay, in this case, I'm just telling you that what we are calculating here is the number of cutting sites we identified at each location. And in this case, for example, here, this part potentially will be just aligned to this region. Let's go into there because this is a taxi. That's why we're doing this. So now let's go here, take a look. Now I'm zooming in. Well, instead of you see this sequencing rates, this signal, this is the Vigo file, and this is the BAM file, and goes directly aligned on top of it. And there is actually a reason for that, because we purposely dented the signal, the read information, and back into the potential cutting site. And in this, so this is a little bit too much for this particular lecture. When we talk about a taxi, we probably will go into that section. And we purposely dented this a few base pair into this read. And because this is a TN5 enzyme protection regions. So although we identify the sequencing rate like this, but instead the cutting site potentially will occur in this location. That's why we put it in at this location here. I can zoom out and then zoom out again, 100-fold. Okay, so now you are seeing there is a strong signal in this region. If you're not really trying to look for the genetic variants in this, I don't think we really need to look at the BAM file. So let's just turn off the BAM file for a second. We can hide the BAM file, and then only the Vigo file will be demonstrated here. You click refresh. It will only give you these two tracks that of the Vigo file. So now you see there's the same genomic loci, and rather than it's only the two strong peaks that are being identified. So what we can do here is, let's see, we zoom out further. So now you can see there is more genes start to show up, and there is peaks here and there in different genomic locations. And specifically, I see quite strong, a lot of genes annotation is around here, and then there is some peaks around these regions. So let's go there, take a look at, take a deep look at. We can zoom in further. So now you see that there is a reproducible signals are being generated here. How to explain that, we'll talk about that in the ataxic section. But as you can see that these are the regions that we can visualize our signal on. We can further zoom out. So now you can see more genes that being picked up here, and that there is a stronger signals around this region. So let's go zoom in this region. Now you are seeing, oh, in this part, this is beautiful. Let's look at it. This part seems this part of chromatin is open, and then this corresponding to a promoter region of this particular gene. And this part, these two are like open chromatin region very clearly in the beginning part of this particular gene. And the same thing happens here. So from all these signals, and after you visualize this, you will have a generic feeling of how well your experiment captured the biological signal. It will be much better feeling comparing to just your information give you a table by the end of the day and just tell you, okay, there is a, here are the list of the differential accessibility. So now you have a much better feeling of whether your data were really captured biological signals. So I cannot spend enough time on this, and these are really fun to watch sometimes. You further zoom in, so you can see this one gene promoter here. There is a very clear signal here. There's another gene promoter and very clear signal. And then you can further zoom in and then to look at the specific locations with the hope whether you can identify some of the footprinting region here. So again, by summarizing, that in these two lectures, or three in the past, I basically went through how to use the Genome Browser to go over to visualize your own data. In summary, there are two elements. One is for small text files, you can prepare the files and directly load into the Genome Browser. And another one is your information will generate a binary format, their specific requirement, but he or she needs also to put that file into a HTTP server, give you the link so that you can get the link and send that into the UCSC Genome Browser. Very similar mechanism can be worked out on other genome browsers like WashU Epigenome Browser, and it's a very well done browser. I really strongly encourage you to look at that, and also integrative genomic viewers that Travis Johnson is going to go over in the later videos. Okay, I hope you enjoyed. I hope you learned something. So I will see you this Friday in the Q&A sessions.